course platform. So have you guys like logged into that platform? Have you guys seen all the various options? So the recording has started. Can any one of you confirm?
Yes, ma'am.
Yeah, that's great. Because in that platform only, we will like you'll be able to see all the recorded classes and the lecture notes that I'll share. And if there and since I have already written in the course handout that this is going to be continuous evaluation, like there will be frequent quizzes. So all those things will be conducted in the platform itself. Okay, so you guys can discuss among yourself if any one of you is facing any trouble in using that platform, and if a solution doesn't come out, you guys can approach me. I'll have a look at that platform and give you the best possible solution on how to use it. Okay, so these the meeting timings, our usual meeting timings will be Friday and Saturday, 5 to 6 p.m. Okay, we can shift the timing if you guys want, like till like dinner time, or like maybe 9-10 p.m. is totally fine with me, but you guys should collectively decide among yourself and then let me know. Okay, and since if you guys want that I share the email or phone numbers, whatever I have contact details, I can share with you guys, because in during this course you will be working on group projects. Okay, so let me know if you need those personal contact details of other classmates, because I feel like not everyone will be joining the sessions, but like some of you will join, some of you will not join, but since you guys want certificates, right, you want to work on this course and gain credits, so for that you need to communicate among yourself. So the basic thing, the prerequisite is you should be familiar with the basic biology, like class 11 12th biology, as well as a bit of cell biology. If you are weak at cell biology, is totally fine, because this tissue engineering field, I'll go into basics of cell biology as well, little bit, so you can catch up. So these are the course content, the topics that we'll be covering. Okay, as you can see, so you can go to your email, you can download this first course handout and keep it with you for reference. Also, we'll be, as I said, we'll be also discussing few research articles, relevant research articles. And what I'll do, I'll give those research articles to you guys. We have to read it. Okay, and then in the next class we will discuss that article and discuss any doubt if you have come across. Okay, and I'll be following the book Tissue Engineering. This, let me highlight. So I'll be following this book uh, by Bernard Paulson and Sangeeta and Bhatia, Tissue Engineering. If you find this book in your library, you guys go ahead and issue that book. If not, I couldn't find any ebook on the internet. Okay, so if one of you is able to find the ebook, please share it with your classmates. Otherwise, the lecture notes are sufficient, I think, because all the notes will be made from this book. And sometimes I'll be referring to the second book as well, Principles of Tissue Engineering. Now, this book you can easily get from the internet. You can download it. And throughout this course, a continuous mode of evaluation will be followed, where there will be frequent quizzes. Okay, and the quiz dates, as you can see, this is the calendar of this course, and the quiz dates are there. So there is no surprise quiz or anything. Don't worry about any surprise quiz. But just the thing of keeping this many quizzes during the course is that you should be familiar with the course content. Okay, you should be continuously going through that notes every week. And the quiz will be of simple MCQ type of format, or one word answer kind of a format. So 15-minute quiz in a class. Okay, so rest 45 minutes will take up the class, and 15 minutes only for the quiz. So this, like this, the quiz will be. And like very small questions will be there. But the thing is, the questions will be of conceptual nature, so you should be able to, if you're like, you should be like completely grabs the concepts that was taught the week prior. If you have any doubts, as I said, you can contact me, you can contact your classmates, you can go through the notes. Okay, so the help is there to help you clarify your doubts. And yes, project for group project, groups will be made. Okay, and one topic will be assigned to each group, And what you have to do, you have to do some literature survey, means go through some research articles on the internet, and like read them, like ऊपर ऊपर से पढ़ लेना, like समझना लेकिन वो research article में क्या है, and try to propose something relatively novel if you think you can, and give submit a 3-page report. That's it. And you have like seen the dates as well, so we will try to stick to these dates. Okay, so but if in between some class gets shifted or something like that, we will manage. Okay, so overall there will be around 34 classes. Okay, and if you guys need any more extra classes for doubt clearing or anything like that, we will also do that. So any one of you can you know volunteer to become the course coordinator. Okay, and create a WhatsApp group. If you guys want that I also want join this WhatsApp group, do tell me or send me the link, I'll also join. Okay, so and at the end of each class there will be attendance. So for attendance you guys have to just you know either you can unmute yourself or type in the message chat box, okay, that I'm present. That's it. So let me begin with the introduction lesson. So this is the first discussion session. So what is tissue engineering? Okay, or if you were to formally define what is tissue engineering, uh, the there is no fixed definition of this field. It gets changed. It is actually changing with the rapidly growing technologies associated with this field. But the first definition that was given in 1988, what it states that it is the application of principles and methods of engineering and the methods of engineering and life sciences. Okay, that is applied towards fundamental understanding of structure function relationships in normal and pathological mammalian tissue, and it also helps in development of biological substitutes to restore, maintain and improve tissue function. So focus on the red highlighted text. So what these red highlighted words are themselves sufficient to help you understand what is tissue engineering. Tissue engineering brings in two things, engineering and as well as life science. So it is not just typical like basic biology that you're familiar with. It needs the input of an engineer. Okay, so when engineer and life science, these two come together, then only a tissue can be engineered. And these engineered tissues or these artificial tissues, if I call another term for that, they are useful in understanding what is the structure function relationship in normal as well as pathological mammalian tissue. So people have created normal tissues, people have created disease models. Okay, so using the principle of tissue engineering only. And these artificial tissues or engineered tissues, they act as a substitute to what? Substitute to normal organ transplants or tissue transplants. Okay, so that these can play important roles like to restore the tissue function or maintain the tissue function, or it can also improve, like a disease, a wound is not healing properly, so for accelerating the wound healing process, artificial tissue can be used. So this is what the entire field of tissue engineering can be summed up using these 4-5 lines. Okay, so this field has grown rapidly, like since 1988 it has grown rapidly because there are so many journals available, okay, that are only dedicated to this. And the regulatory standards, like if you want to submit any patent or anything like that, so for judging those patents, new regulatory standards are coming out. Okay, and there is hundreds of articles that has been published so far in the field of tissue engineering. Now if you see this particular bar diagram, you can like understand what I have just said so far. So if you see in 2021, the market size, tissue engineering market size is around 13 billion US dollars, and by 2013 it is expected to grow to 31 US billion dollars. So like more than double the growth is expected or predicted, because this field is like such an important field. It has a large potential to impact human life, and as population is growing, more diseases are coming up, right? So here tissue engineering, where it doesn't require any human sacrifice or anything, you're artificially making an tissue. So for so these artificial tissues can be used for healing, studying like various kind of diseases, etc. Like it has a huge potential. So in India there are no yet there is no tissue engineering companies, but in abroad there are companies who are only making artificial tissues, and they are supplying it to hospitals and etc. But in India, it is yet to happen. Like big pharma companies like Biocon, Doctors Reddy and all, they are slowly transitioning towards making artificial tissue models. Till now they are like the market, capturing the entire drug discovery and drug delivery market, but slowly they are going to shift to tissue engineering, and hopefully one of you will become the next generation tissue engineers and get a job there. Okay, so as I told, I have not told, so I'm going to say that tissue engineering is an interdisciplinary field, meaning it doesn't only require the participation or the knowledge of basic biology, but it also requires inputs from other areas of engineering as well, like material science engineering, where people study about various kind of polymers, etc. Then cell and molecular biologists, they can also, their knowledge can also play an important role in developing the field of tissue engineering. Then immunologists, the policymakers who make the laws or set the regulatory standards, and chemical engineers like me. Okay, and surgeons, definitely the input of the doctors and the surgeons. So when all these people from various fields, when they come together, then only artificial tissue, a successfully marketable artificial tissue can be engineered. Okay, so if you guys have any doubt at any point of time, feel free to unmute yourself and ask questions. Okay, I won't mind at all, don't worry. So conventionally, if you see, in tissue engineering or artificial tissues, what we do, we use cells. Okay, we do not use their proteins, we do not use their genes, we use the cells. Okay, so that is why it is called cell based therapy. Okay, and cell based therapies, or this and they are not very new. Okay, it has been used for so long. So people use whole organ transplants, right, which are biological implants, or like when anyone's bone is fractured or anything like that, shattered to pieces. So they use rods and etc. Or they use artificial prosthetics, like people wear it. Okay, so when any limb is lost, so these kind of treatments or therapies are there. And when people are using cell based therapies, here they are using live cells. Okay, they are not using dead cells, they are using live cells, which are itself are the therapeutic agent. And the goal of using live cells is that we are going to harness its elegant biochemistry machinery. Okay, and allow these cells to produce the required drug or therapeutic protein inside the body. And sometimes when we want to cover a surface, okay, or like a skin burned area is there, we want to cover it, so we want large number of cells to come there and sit on it. So in that case we are actually harnessing the cells replication ability. So that this is why people are moving towards cell based therapies, and why cell based therapy is very much useful. So as I said, cell-based therapies are not new. There are a few existing cell-based therapies, like there is blood transfusion, which you are all familiar with, right? Then platelet transfusion. So in it happens in patients who have blood clotting defects. Then there is bone marrow transplantation, where the entire bone marrow is transplanted. We will come to it a little later. Then there is hematopoietic stem cell transplantation. So these are all like existing, very well established, clinically relevant cell-based therapies. now there can be various sources from which a doctor or a tissue engineer can get their cells from. So it can be autologous. Okay, by autologous I mean you take the cell from a donor and then give the cells back to that person only. That is called autologous. Then there is allogenic. What is allogenic? Here what happens, you take the cells from a donor, like you take the cells from me, and my doctor is going to take the cells and give it to my sister. So we both are from same species, but the person is varying, so that is allogenic. And there is syngenic. So here what happens, the recipient and the donor, they are homozygous twins. Okay, so they are genetically identical to one another. So one identical twin, identical twin is giving her cells to another, like another sister, his or her sister or brother. So that is syngenic. And there is xenogenic. Here what happens, you take the cells from a pig or a cow, okay, or a different animal itself, and you transplant it into humans. That is called xenogenic. That is cross species. So cells are derived from an animal that is not belonging to our species. It is belonging to some other species. But please note that people are actually around the world, there are few labs around the world who are trying to develop universal donors, universal donor cell lines. So there are universal blood types, right? Means AB group, AB group people can donate blood to anyone, okay, with any blood type. Similarly, people are trying to develop universal donor cell lines or cell sources, so that people don't have to worry about finding a donor. Like in allogenic, you have to find a donor person who is willing to give you cells, a lot of cells, not one or two cells. I'm talking about billions of cells. So for that, people has to undergo surgical intervention and all. So to overcome that problem, people are trying to make universal donor cell sources. So people are not yet successful. The research, a lot of research needs to be conducted before it can come into the market and people can use it commercially. So far I hope it is clear to you guys. If there is any doubt, please let me know, or else I'll continue. No doubts? That's great. So if you have any doubts, please let me know. Okay. Otherwise, the content so far is relatively easy and easy to understand. So I'll move fast. Okay. Now, let me introduce to what is called tissue engineered medical products, or in short TEMPs. So these are the products which are made by using tissue engineering principles. So these are newer cell-based therapies. These are not like the existing cell-based therapies that we talked about, like blood transfusion, etc. These are not that. These are the newer cell-based therapies that incorporate significant cellular manipulation in vitro. So here in these products, we culture the cells first, and then we incorporate the cells on some kind of a support. Okay, and so we are basically manipulate. We are adding some human manipulation before that product is ready for its clinical use. So there are a few examples of this, like people are using chondrocytes for cartilage repair. So it is called autologous chondrocyte transplantation. That I'll come to it later. Then there is people are actually growing liver and kidney cells in extracorporeal support devices. So these are devices that are present outside the body, but through tubes they are connected to your body, and they are able to give some kind of a important function like liver function or kidney function. So these are called extracorporeal support devices. Then encapsulated beta islet cells for diabetes. So people are encapsulating beta islet cells, which are the cells responsible for secreting the hormone insulin. So these encapsulated cells are proposed as a treatment for the patient, diabetes patients. and people are also making sheets, like planar sheets of dermal fibroblast, which can be transplanted on ulcers or burns, okay, and promote healing. Also, people are genetically modifying the myocytes, the muscle cells, which are used for the treatment of muscular dystrophy. So these are some of the examples of the tissue engineered medical products. So as you can see from this example, that they are of various types or several types. So a tissue engineered medical product, or TEMPs in short, I'll refer to them as TEMPs. So these products, they can be of hybrid nature, means with cells as well as some non-biological component like a polymer construct. Okay, so one of you have said some uttered a term called scaffold. So polymers are used for making scaffolds or external support for the cells. So these kind of hybrid products can be made. Okay, and they now these are called TEMs, or these products are such that their role is to induce some kind of an immune response. Okay, so for example, regeneration. So here people are using stem cells, okay, to promote a tissue regeneration, or if I get too ambitious, then organ regeneration. So things like that people are doing. And also the cells are genetically modified in vitro, means in lab scale on a petri dish, they are modified genetically, and then these cells are used. So one of you again said the term IPSCs, right, induced pluripotent stem cells. So when you're using IPSCs for making some kind of a tissue engineered medical products, so that is also one possible way to go. So till now, if there is any term that you're not familiar with, okay, and I have just like said it on with flow, let me know. I can explain that term to you. Okay. So if any queries, let me know. Now, the ability to reconstitute tissue function and produce stems, that is tissue engineered medical products, at clinically meaningful scale, it has a wide spectrum of application. So you have just seen how these stems can be beneficial for the treatment of diabetes, the muscular dystrophy, etc. So it has a wide spectrum of application. Effective treatment or cure of several tissue deficiencies will greatly improve the quality of life of affected individuals and make them more productive, and like so that they can contribute to the economic status of a society. So it has that is why as you saw, tissue engineering market size is increasing, and it is predicted to be increasing like more and more. Okay, so it has a huge impact. It can have direct affected people. Okay, and if you are wise enough, you can also create products at a very low cost. So that is also a thing to be kept in mind if any one of you is like interested in future to make a product of yourself and then market it. Now if you see this particular table, I've taken from the textbook tissue engineering. So here this data is from 1993, I think. Yeah, 1993. So here it is showing how many procedures, like surgical procedures, or how many patients are there of each kind of a particular disorder like skin burn or joint replacement, okay, per year. In this is the data in United States. So you can see per year around 2 million people, 2 million people are victims of burns. And when you see the joint replacement statistics, there are around like 55 lakh people who are in need of this kind of procedure, and there are several others. So these data itself shows like why one needs to create an artificial tissue, like the artificial skin or bone grafts, okay, tissue engineered bone grafts, how these can be very much helpful for patients worldwide. Not only patients, it will make the job of surgeons who are particularly performing these procedures, it will make their life easy as well. And when you have a look at this particular chart, this chart shows how the demand for organ has increased rapidly. And this is a 6-year data from 1988 to 1994. Okay, so you can see from 1988, the total number of patients who were in need of a organ or tissue transplant is 15,000, and by 1994 it has almost doubled, right? It has like grown to 35,000. This is just in 90s, like these two data from 90s. Now when you think of how much population has grown till 2023, so you can definitely imagine these data have rapidly, like exponentially, has doubled or tripled. Okay, so that is why this field is like taken up as a research interest in more and more labs in India as well as abroad. Okay, because it has a lot of potential to help the people who are in need. So let me give you or explain to you guys few examples of TEMPs or tissue engineered medical products. Okay. So first one is the procedure that is called autologous chondrocytes transplantation. So here, let me like briefly go through the points. So cartilage is a tissue. Okay, you guys know it is a connective tissue, and it is avascular in nature. Means it does not have any blood supply to it, connected to it. It also lymphatic, means it does not have any lymphatic vessels connected to it, and it is aneural. That means there is no nerve ends. And this tissue mainly consists of extracellular matrix, and it is actually collagen. Okay, extracellular matrix is made up of collagen. It is a natural polymer. And the chondrocytes, they are there, but the cells are very low in number. Okay, they are scarcely dispersed in the extracellular matrix. Now for deep cartilage, defects in the knee, uh, people have come up with autologous chondrocyte transplantation method, where what happened, a small biopsy is collected from the patient's knee outside the affected area. Okay, so if a defect is or a wound is there, you have to collect the biopsy of a healthy tissue which is bit far from the, not too far but bit far from the affected area. And then after have taken the biopsy, you have to liberate the chondrocyte cells by digesting the ECM or extracellular matrix by using enzymes like collagenase, etc. And then you allow the chondrocyte cells to grow, okay, in cell culture process. So once the cells have grown to like millions of, in million number, then you harvest those cells from the culture and reintroduce the cells into the patient where, but at the affected area. So if you see this particular diagram, so here the patient is having, this is the affected knee part, which is having the lesion or the wound that needs to be treated. So biopsy you need to collect from the healthy cartilage, means not too far from the lesion, but from a normal area of the cartilage. You have to first collect the biopsy. So people use punch biopsy, okay, for very small amount of tissue to get out, okay. But you have to make sure whatever wound that you are creating for collecting this biopsy, it should not be too big that it cannot heal on its own. That's why a small biopsy sample is taken. Then you doctors enzymatically digest it, means remove the ECM and liberate the chondrocytes. These cells are then cultured in tissue culture flask, okay, for 11 to 21 days. And then you do trypsin treatment, okay, to liberate the cells. Trypsinization it is called. And then you get around millions of cells, like 2 to 5 millions of cells. Now this cell suspension, it is present in a cell suspension in a media, cell culture media. You take this cell suspension in a syringe, okay, and inject it directly on the affected area where there was the lesion, okay. And they also suture some kind of a protective flap, okay, or you can simply put some kind of a polymer, biodegradable polymer over it, suture over it. Why? So that the injected cells do not flow out. You want to trap those cells, and it should be trapped for enough time so that the cells can adhere to the lesion and multiply, and promote wound healing. So that is why a flap is sutured over the lesion. So this is you are taking the since you're taking the cells from the patient and then injecting back to the patient only. That is why it is called autologous chondrocyte transplantation. This is a very well established technique, okay, that people are currently using in clinical setting. Is there any doubt so far regarding this technique? No doubts?
No, ma'am.
Yes,
Yeah, yeah. Go ahead. Go ahead.
ma'am. Like you said that in this image that it is cultured in the rocks bottle, and that it will be how much time they are passaging like to to grow in this media, and after that is the cell is able to redifferentiate itself into the knee area itself,
So,
like we
hmm,
when we
yeah.
cultured multiple times of the cells, they can be form a cell line,
Mm-hmm.
and it is maintained like 11 to 12 days. It means that it will take 3 to 4 passage, even more. So it is more likely to be a cell line, or it will be the same as the previous one.
Usually when we culture primary cells, so here the chondrocytes, they are termed as primary cells because they're taken directly from the patient. So when you do culturing of primary cells, you do not passage it that much. Okay, so usually they arrest, their growth stops at around 3 to 4 passages. Most of the primary cell lines. So which is why when they collected the biopsy, the cell number should be such that it should within 3 to 4 days it should cover. Okay, and it also depends on the cultivation time. So here they have written 11 to 21 days. So it is possible. It is possible for the cells to only one passage, within one passage. That passage will take 11 to 21 days for the entire flask to cover and form a monolayer. So it can happen for primary cells because the doubling time will be different. Okay, so we will talk about the doubling time of the cells as well, and it varies from each cell type. So it is not going to become any cell line. Cell lines, people do some viral transfection, or they modify their gene so that they can go up to 100 of passages. But for primary cells, we restrict to 2 to 3 passages only, because that is when the cell activity is the most. Now since we are going to inject it back to the patient, we are not going to passage it that much, only 1 or 2 passage, till we get millions of cells, and then directly inject it back. Okay, is that
Thank you.
clearing your question, or if you have any more doubt?
Yes, I'm clear.
Okay, great. So no more doubts. We will go and look into another type of TEM, or tissue engineered medical product. So this is called extracorporeal hepatocyte based bioreactors. So extracorporeal means, the term means outside the body. So you are making a small bioreactor which is going to be connected with your body. Okay, and but it is going to be placed outside. So the patient has to carry it with him or her. So these kind of corporeal devices can be made for liver or kidney, etc. So here I'm going to talk about the liver organ. So liver is a very important organ, as you guys know. So it can perform a diverse area of vital functions like metabolism. It is responsible for metabolizing protein, fat, carbohydrates, then detoxification of foreign products as well. Then it produces various serum proteins like albumin, then clotting factors, and it is also responsible for production of bile. The bile juices help in digestion. So it is a very, very important organ for our body to survive on. Okay, so if anything happens to it, like chances of survival, becomes very slim. So now, which is why, and since this is a very complicated organ to engineer, as you guys can think, so there is no single pharmacologic, prosthetic, or surgical therapies there to treat any liver disease. except for whole organ transplantation. So if you get a donor who is willing to give his or her liver, so then only that patient can survive. Otherwise, only one or two drugs going to sustain his life for a few months, and then probably he'll die. So as the wait list for organ, liver whole organ is increasing, so what surgeons are doing, they are splitting the liver into two. Okay, and so if they get one liver organ from any donor, luckily they split it into two and give it to two separate patients. And why it is possible? Because liver is known to have enormous regenerative capacity. So a small portion, one half of the liver, can with time or over the years, it can form the entire liver. This liver cells inside the body, they are very much uh, fast in proliferation, and hence the enormous regenerative capacity. Now, when the use of this kind of bioreactors or the devices comes into picture, so these devices, they have been proposed to stabilize any patient, okay, until a whole liver organ becomes available, or when the transplanted organ becomes fully functional. So during those temporary periods of time, these devices can sustain the patient, okay. Uh, so it is not a permanent therapy, as you can understand. So how these devices are made? So these devices basically consist of immobilized animal or human hepatic cell line. So here cell lines are used because now these cell lines are not going to be in direct contact, means they are not going to be present inside the body. So you can use an animal cell line as well, which will not show any immunogenic response. So these cell lines are used and they're immobilized. They may be immobilized inside a hydrogel material like collagen, etc. So that and these cell lines should have the ability to provide all kinds of liver functions, means synthesis of any kind of therapeutic proteins. So these cell lines should show those kind of characteristics for them to be used in this device. So patient's blood or plasma, it is flowed through the device and it is brought in contact with the immobilized cells. So when it is brought in contact, this immobilized cells, they will secrete out the metabolites, important metabolites into the blood or plasma, and it will be then passed into the patient's bloodstream. So you are using only this device for the important liver metabolites that the person needs, right? So for metabolism of liver, protein, fat, etc., you need some kind of enzymes. So these cell lines are able to produce the enzymes and then provide it, secreted into the patient's bloodstream, and it goes into the patient's body. So how a typical this is how a typical device looks like. So guys, am I audible to you? I think I temporarily lost my what do you say, my connection.
Audible, ma'am.
Yeah, audible, no? Okay, fine. So this is the image of how the extracorporeal liver device looks like. So here it is one type of configuration. If you look up in the internet, you can find many configuration that people have made. So here there are hollow fibers, okay, hollow fibers within which using collagen gel, they have immobilized hepatocyte spheroids. So if you have cultured any kind of hepatocyte cell line like HepG2, they have a tendency to form spheroids easily. Like in T25 flask or T75, they will easily form means clusters. And when you use like hanging drop method, if you have heard of, you can form spheroids. So here spheroids are embedded into the collagen gel, and this is what is done in each hollow fiber. So this hollow fiber configuration increases the surface area. Okay, so and the both ends, there is one end for the patient plasma to go in, one end for patient plasma to come out. And there is flow of the plasma, as you can see. Extracellular stream is there, which is flowing outside the hollow fiber. Okay, and there is stream that is flowing just close to the cells, that is intraluminal stream. So with in between the hollow fibers, you can guess there is some space. So through that space also the blood plasma, patient's plasma or blood is flowing. So this is how the flow is engineered in this kind of devices. And immobilization is very much important so that your hepatocyte cell line doesn't enter the patient's bloodstream. Okay, so if you are using animal cell line, so it can cause you, as you can guess, immune rejection or some kind of complication. And since these devices are used for those patients who are themselves recovering from an extreme organ transplant procedure, or they do not have a functional liver themselves, so to those patients you have to be very much careful while designing this kind of devices. So these kind of devices have been proposed, and I don't know whether they're commercially used or not. Maybe you guys can check it out. But I think similar kind of devices must be there. Okay, so we will quickly go through another type of TEM example. Okay, and then I'll wrap up today's session. So if at any moment of time I'm not audible, please let me know. Okay, because I think there is some net issue from my end. Okay, so another type of TEM is where they're encapsulating beta islet cells. Okay, so beta islet cells are the cells which are present in pancreas, and they're responsible for secreting the hormone insulin. So pancreas in many insulin dependent diabetic patients, they have lost their ability to secrete insulin. So in diabetes, type 2 diabetes patients, it is the like end stage. Like towards the beginning some insulin production is there, but if it becomes an extreme scenario, insulin production absolutely ceases. So in that scenario people has to inject insulin extravenously, right? They have to inject insulin externally. But here what they're suggesting, why not transplant beta islet cells itself inside the patient's body, so that the cells inside the body, they will start producing the insulin directly into the bloodstream. We don't have to worry about buying insulin, which is quite costly, and then injecting into the body. So in these cases, cadaverous pancreas, means pancreas organ taken from the dead body, from some dead body. These cadaverous pancreas, they can become allogenic source of islets of Langerhans, which contain the insulin secreting cells, beta islet cells. So you take the beta islet cells from a cadaver, okay, and use that uh, and encapsulate it in some kind of a polymer, and then inject it into the body. So they inject it into the liver portal vein, and once these islets, they are lodged into the liver. Like liver portal vein is that vein that connects to the liver and gives it blood supply. So there they want the beta islet cell to be sitting there, and want cells to secrete insulin, so that in the liver metabolism of glucose can happen. But here in this example, they are not using any kind of a barrier for the immune cells like the T cells, B cells, or the antibodies to recognize the foreign cells. So because of which this kind of grafts, they get rejected, and the therapeutic benefit is only temporary, which is why people have proposed to encapsulate these beta islet cells so that they are isolated from the body's immune system. The antibodies or the antigen presenting cells, they should not be able to recognize these allogenic islet cells as foreign, and allow them to secrete insulin directly into the bloodstream. So here separation between the islet cells and the body's immune system is made in such a way that antibodies are not allowed to enter or get in contact with the allogenic cells, but the separation is porous enough for the nutrients and the insulin to pass through. Okay, and there should be a continuous exchange between nutrients and insulin between the bloodstream and the encapsulated beta islet cells. So these are some examples of how encapsulation is done. So it can be done in a fiber form, where within the core of the fiber there is the beta islet cells. So here spheroids of beta islet cells are shown, and the the shell of the fiber, it is porous enough, as well as it is acting as a barrier for the antibodies or the macrophages, etc., to get in contact with the allogenic cells, but it is allowing the exchange of glucose and insulin to and back from the islet cells. Okay, so this is one configuration. Another configuration is you can use microspheres. Okay, microspheres or nanospheres, this kind of spherical configuration, within which within center of these spheres there is the spheroid of beta islet cells, and again the shell is such that it is allowing the transport of both glucose and insulin, and it is functioning well. So these kind of so there are a few more examples of TEM. So for today's class I'll like stop, and I'll continue in the next class, that is on next Friday. So if there is any doubt, please let me know, and then I'll take your attendance, and we'll wrap up. Is there any doubts so far? I'm not able to understand whether it's my network issue that you're not able to hear me, or if there is no doubts at all, because as I told you, there is no current right now, and the weather is also not good in Kanpur. So can any one of you respond that I'm audible or not?
Yes, ma'am, audible.
Yes, ma'am, you are
Audible.
audible.
Great, great. So there are no doubts from your end?
Ma'am,
Yeah.
ma'am, there are three papers. I mailed you about that.
Yeah.
Like dates are like 7 July. Paper is postponed already. Like three dates are there, which is overlapping with the timings, because my shifting of paper is 3 p.m. to 6 p.m.
Okay.
So 20 July, 22 July, and 5 August. So these are coming on Friday and Saturday. That's why I'm overcoming your session.
Means it is overlapping my session. Okay. So you said
Yes,
that
ma'am.
it you will be able to join from 7:30 to 8:30, right? During those days.
Yes, ma'am. Like, yes, ma'am.
Okay. So I ask rest of you, if during the dates of Janvi's exam, would you be able to join the session, like my session from 7:30 to 8:30? Would you guys be able to do that? Or is there is any other commitment from any one of you?
Yes, ma'am, it's totally fine because I am also having the same date examination or the same timing. Because Janvi and we both are giving examination at that time.
Okay, okay. So what about rest of you? No issues, right?
No issue, ma'am.
Oh, great, great. So Janvi, during the dates of your exam, um, can you repeat the dates so that I can note it down?
Yes, ma'am. 28 July,
28 July,
22
then
July,
22 July,
5 August.
5 August. So in these 3 dates, we will have our session from 7:30 to 8:30 p.m. Okay, I'll also drop a official mail to you guys, so that you have a like you can keep it in your calendars. Okay, so that time is shifted, so your problem is sorted. And I wanted that one of you guys should volunteer to make a WhatsApp group. Okay,
I'm
and
actually,
yeah, yeah.
ma'am, I'm not able to chat, like, please the group link here. Can you enable?
Yeah, yeah, can you repeat? I couldn't get what you just said.
Ma'am,
Can you repeat?
the chat option in this session are not able to do.
Okay, so
Ma'am,
you, yeah.
she's saying that she's created the WhatsApp group and is unable to send the link in this meeting. So please allow her.
Okay, okay. So you're not able to access the chat option, no? Is there anything like Q&A? Can you option the access the Q&A section? Janvi, are you able to access the Q&A session? Or maybe I'll do one thing. I'll share the contact infos, okay, in an email with all of you, okay, so that you guys like Janvi can it's herself email you guys, okay, and email the WhatsApp link. Is that fine?
Okay, ma'am.
Okay, okay. So that's great. Like Janvi has already created a WhatsApp group where we can communicate. And so let me just quickly note your attendance. Then we'll wrap up.
Or either we can share our mobile number here.
So you guys, but Janvi is saying that chat option is not accessible to you guys.
But we can speak our mobile number.
Uh, you can, it's totally fine. If someone is noting down, you guys can share your mobile numbers, but I would say that that mail option you can look into later, and you can do it later actually through via mail, since Janvi will share the WhatsApp link, right? So the mobile numbers will automatically get added in your WhatsApp group. So then that will be fine, I think. Okay. And also not all of you have joined. There are many more who have not joined, so for them also it will be beneficial. Okay, so just quickly one by one tell me your names, like so that I can note it down. So Nidhi Priya is there, like let me just note it down. Okay,
Yes,
Nidhi
ma'am.
Priya, yeah, Nidhi Priya is there. Kirtan Kumar is there, right? Yeah. Let, okay, next, who is next? Please tell
Ma'am,
me your
Bhupendra sir.
Bhupendra. Okay. Okay. Next,
Janvi Sharma.
Janvi Sharma. Okay. Next, anyone else is remaining?
Yeah.
Uh, can you repeat?
Aditya.
Aditya. Okay. Thanks, Aditya. Aditya Bhatt. Okay. Anyone else left? No, right? Everyone is here.
Ma'am, have you noted my name, Harshita?
Okay. Harshita. Great. So thanks all for joining today's session, and I'll see you all next Friday. Okay, you guys can leave the
Ma'am,
meeting. Yeah, yeah.
ma'am, actually, I already have the account in the iionline.com.
Mm-hmm.
Okay, okay, so in that mail it is mentioned that you have the already have the account. So please log in. I have logged in, but in your course